Hi, everybody. Welcome to another edition of the Australia-Israel Scholar Seminar Series. I'd like to first acknowledge the Jagera people and the Turrbal people. They're the traditional custodians of the land on which we gather today. We pay respects to their elders past and present. We extend that respect to Aboriginal and Torres Strait Islander people here today and to Indigenous people and people throughout. It's a great pleasure to have Dr. Vered Irihimovitch from the Vulcani Institute speaking to us today. She's speaking from Israel, and she'll speak about molecular events involved in fruitlet, abscission, and mango. Very much looking forward to your talk. Thank you, Vered. All yours.
Okay, so good evening, everyone. So my name is Vered. I'm working at the Vulcani Center in the Department of Fruit Tree Science. In my lab, we're working both on avocado and mango. And today I want to share with you in this short talk some of the results that we got over the years in studies that we did in the lab, focusing our attention on trying to decipher molecular events involved in fruitlet abscission in mango. So as you might know, in mango, like in other subtropical fruit tree species, despite satisfactory flowering and fruit set, its production does not always meet its potential, and this is due to, among other factors, intense natural fruitlet and fruit drops. So in general terms, mango fruitlet shedding take place of fruitlet and fruit shedding take place during 3 stages of fruit development. The highest abscission rate are observed in the first 2 months after flowering and fruit set, next followed by the mid-season drop and lower abscission rates that are observed just before fruit reaches maturity. Now, in our studies, we're focusing in trying to decipher what is occurring during the onset of the mid-season drop. And it's important to note that especially this wave of drop of fruitlets aggregates during a heat stress condition, as we are facing in the last year, special very warm summer, which leads to an increase in the grower incomes and therefore constitute an agronomical problem. So when we're talking about the abscission, a shedding of an organ from any organ, not only fruitlets, from the main body plants, it is quite acceptable from studies in model organisms that it is conducted by a sequential set of events, which is depicted in the following scheme. So we're starting by phase 1, which is differentiation of an abscission zone. Phase 2 is a phase in which there is an acquisition of these cells in the abscission zone to respond to stimuli that induce the abscission. Phase 3 is the phase of the separation itself. There is enlargement of the cells in the abscission zone, and there is also an increase in their cytosolic pH. And there is activation of the process by activation of different cell wall degrading enzyme, which leads to the separation of the organs from the mother plant. And in the last step, there is a formation of a protective layer in the place where there was an abscission. Now, 2 main hormones antagonistically control this process. These 2 main hormones are ethylene, which acts as an inducer, and auxin, which in low level acts as a repressor. In general terms, diffusion of ethylene from the organ that is about to shed is accelerate abscission by activating cell wall degrading enzyme at the abscission area, and by contrast, auxin flux from the about to shed organ to the through the abscission zone inhibits the abscission by reducing the sensitivity of this zone to ethylene. Another key component that influenced this process was discovered in Arabidopsis in model plant. It is called IDA, inflorescence deficient of abscission or in abscission. It is a small secreted peptide which acts as a signal molecule, which also initiate this process. And it was suggested that its it's the production of IDA or the upregulation of IDA occur in an ethylene independent pathway. However, this concept was recently challenged, and I will talk about this further. So when we look at the pedicel abscission zone in mango, we're using cat fruitlets. We can see that the mango pedicel abscission zone is already pre-differentiated. It is organized by a regular roll of small cell. And if we induce abscission by the utilization of ethaphone, which is an ethylene releasing compound, this is how it's looked under the microscope. And we can see already following 48 hours, the separation process. So what we knew about mango fruitlet abscissions when we started or during the year, what was known about this process. So not surprisingly, it was found that mango fruitlet drop correlates with enhanced endogenous ethylene production, both in the seed and in the pericarp tissue. Next, studies from our labs and other labs shows that the process of abscission in mango is associated with upregulator of specific ethylene receptor genes. And it was also interestingly shown that mango fruitlet drop is associated with reduction of the transport of auxin, polar to auxin transport, from the fruitlet through the abscissions. So keeping this in mind, in a study that we performed a few years ago, to try to decipher whether a mango fruitlet abscission is occurring in association with activation or deactivation of specific auxin-related genes. What we did, we used a mango fruitlet explant. We went to the field, we collected the explant-bearing fruitlets, and in the lab, we either treat them or not with ethaphone. And then look at the expression of different auxin-related genes. So we noticed that following an increment in ethylene production or ethylene release, there was downregulation of genes that encode for auxin synthesis, downregulation of genes that encode for auxin transporter, downregulation of genes that are responsive to auxin, both in the pericarp and in the abscission zone. By contrast, we saw that there was upregulation of GA3.1, which is a gene which encodes for the formation of auxin aspartate, auxin amino acid conjugate, which leads auxin for degradation. All this together leads to a reduction in the auxin level, reduction in the polar auxin transport capacity through the abscission zone, and eventually to the shedding of the organ. So this was one of the studies that we already published. And next, we want to focus our attention to see whether in mango there is also a possibility that IDA protein fulfill a role in the abscission process. Few words about how it's operated in Arabidopsis, which is a model plant. In Arabidopsis, IDA is in charge of the abscission of the floral organ. It is a small secreted protein that once it recognized by a specific substillazes, it is processed and the small peptide is recognized by a specific receptor inducing. Following this connection, there is an induction of a cascade of event which leads to the activation of cell wall remodeling and degrading enzyme. So when we start looking at whether or not this exists in mango, we knew that they may assume similar role in fruit trees because there were studies in citrus and in lychee claiming that IDA also take part in the abscission process in these fruit trees. Yet there was no information regarding IDA from mango. So to start looking about whether IDA can control the abscission process, we first identified two genes. We named them Mangifera indica IDA1 and IDA2. We saw that by blasting them and by aligning them that they possess conserved domains that are necessary for the function of the IDA. And next, we want to see whether they are somehow involved in the control of mango fruitlet abscission. So for that, we did experiments, again, using both mango fruitlet explant and mango fruit-bearing trees. in which we either treat them or not with ethaphone, which is the ethylene releasing compound, in the field, and we look first at the abscission rate. So as you can see, following treatment within 24 hours, there was an increment in the abscission rates, which reaches 100% rate by 3 days in the fruitlet bearing explant, and in the control, the abscission rate were significantly lower. In the fruitlets bearing tree, there was a similar phenomena. Here, the abscission rates reaches 80% by 7 days and then remained almost constant under control condition. There were also an increment in the abscission rate, yet they were significantly lower than in the treatment of ethaphone. So this is the natural drop, actually. So we then took the abscission area of the fruitlets. We collect them prior to the peak in the abscission in both systems, and we look at the expression of IDA1 and IDA2. Both genes were upregulated in the fruit-bearing explant by 12-hour and 24-hour following the treatment, and here by 24-hour following the treatment, IDA2 increases and IDA1 increases and then decreases and was significantly higher than under control condition. To try to correlate between the IDA and the expression of a cell wall modifying gene, we look at the expression of 3 pectinolytic enzyme encoding genes, and we saw that their expression is somehow correlative with the upregulation of IDA2. And we also look at the cell wall loosening protein encoding gene expression, expansin, which is expression patterns correlate with the expression pattern of IDA1. To perform an internal control, we also look at the expression of different ethylene signaling genes, which of course also were induced following the ethaphone treatment. So taken together, these results suggest that indeed IDA can take a place in regulating fruitlet abscission in mango. To try to see whether IDA from mango operates like IDA from Arabidopsis, we overexpress the gene from mango in Arabidopsis and have several lines that were in which the gene of mango was upregulated in the Arabidopsis. And when we examine the flower at different positions along the inflorescence, we were able to see that when we overexpress IDA1 and IDA2, there was an early abscission of the floral organ in position 5 and 6 as compared to control and wild type that occur usually at position 8 and 9. We also use a pH sensitive dye, which become green when the pH increases, and we were able to see under focal microscope that indeed abscission of floral organs, the pH become more alkaline earlier than in wild type plants. To wrap up the story, we also overexpress both genes in the background of the IDA mutant from Arabidopsis. In the IDA mutant from Arabidopsis, there is no abscission of the floral organs, and we were able to see that by overexpressing it in the background of the mutant, we could restore the abscission ability. So if I quickly summarize up to here, our data provide different lines of evidence supporting the conserved function of both IDA1 and IDA2 for mango in regulating mango fruitlet abscission, and also suggest that the putative mango IDA and its receptor-like module operates in this case, in an ethylene-dependent manner, because we spray with ethaphone and we increase their expression. Furthermore, our results also indicate that both genes function in a similar mode as IDA from Arabidopsis to promote organ abscission. In the next step, or what actually we're doing right now, we even want to try to get a more deeper understanding of what is occurring during fruitlet abscission, and we did a transcriptomic analysis using control and ethaphone treated fruitlet abscission zone, and the results are summaries in heat map. In general, when there is color of red, it's upregulation, blue is downregulation. So we saw that there is an increment in the ethylene response gene, downregulation of some auxin-related gene, and even upregulation of ABA encoding gene, which are also inducing abscission. We noticed, of course, that following 48 hours of treatment, there was an increment in the IDA gene as compared to control. And interestingly, what we also noticed, of course, of the cell wall modifying genes also increased. And interestingly, we saw that there was also upregulation of genes encoding for precursor of phytosulfokin. What are phytosulfokin and how they are related to our story? So phytosulfokin, a small peptide hormone, recently suggested to act as new player affecting drought-induced flower abscission in tomato. They operate in a similar fashion as IDA. They are small secreted protein that once they are cleaved, the small protein is recognized by specific receptor, which induce a cascade of events, which leads to activation of the abscission process. And we indeed were able to find that there are 3 phytosulfokins and 2 phytosulfokin receptor for mango that are upregulating following the treatment. And we are now trying to see how they operate and how they control abscission in mango. Especially, we're interested to see how they might control the abscission process under heat stress condition. Another interesting feature that we got and saw in the transcriptome analysis, and this is the last thing that I will discuss on from the transcriptomic data, is that there is also an increment in the genes that encode for reactive oxygen species generation and reactive oxygen species response. It is something that is known also from the abscission of other from studies in other fruit tree species. So we thought that in this case, it might be interesting to see whether we can use a spray a compound that might reduce the formation of the reactive oxygen species in order to mitigate or to reduce the abscission. And in here, I'm doing just a thematical pause to introduce you with hydrogen sulfide. Hydrogen sulfide is a molecule that serves as an important plant gaseosus signaling molecule. It has been recently documented for its protective effects against different biotic and abiotic stresses, and it increases the antioxidant activity. And it was also recently shown that, at least in tomato, hydrogen sulfide treatment or sodium hydrosulfide as a substance used as a donor of hydrosulfide suppress the shedding of the tomato fruits and flowers. So we decided to look whether they can suppress also the shedding of the mango fruitlets, and we perform experiment in our in vitro system using both Kent and Shelly cultivar. Shelly cultivar is an Israeli cultivar, and what we were able to see is that indeed, when we did a pre-treatment with sodium hydrosulfide and next we induced the abscission with the ethaphone, we were able to reduce the abscission that is occurring due to the ethaphone treatment, and we were reducing it almost as the control. We look this time, we use a fluorescence dye, which is a Ross marker. And what we were able to see is that with 48 hours, there was no accumulation of this marker in the abscission area under control condition. With ethaphone, there was a high increment apparently in the Ross accumulation in the abscission zone. And when we pre-treated the explant with sodium hydrosulfide and next with Ethaphon, there was no such intense accumulation of frost in the abscission area, which correlate with the reduction in the abscission itself, in the rate of abscission. So this seems really interesting, and nowadays we are doing an experiment in the field in which we are trying to spray with sodium hydrosulfide to see whether we can increase the retention of the fruitlet in the onset of the mid-season to, of course, increase the incomes of the growers and get higher yield. And with this, I want to finish this short talk. Just to mention, give the credits. These are the guys that work in the lab, and most of the results that I show that are from performed by studies from Avinash Chandra, which was a postdoc in the lab. The work is done with collaboration with Yuval Cohen, Sonia Filosof-Hadas, Simon Meir from the Volcanic Center, and also with Gad Erkin from the Tzemach Station. And we're working, the trees that we're working and doing all the experiments are trees that are localized in the north part of Israel, in the North Galilee. We can see from here a nice view over the Kinneret Valley. It's one of the most nicest places in Israel, and it's really nice to walk in there. With this, I will finish. Thank you very much.
Thank you very much. It's a small audience today, but in the name of those that will watch this on YouTube as well, I want to thank you. Can I ask a question or two?
Yeah, you can.
So thank you. So look, this is an outsider question. I'm not an agronomist, more a statistician, but I'll first kind of a naive one. So when you look at the genetic markers associated with Abscission.
Sorry, can you speak louder?
Oh, yeah, sorry. So when you look at the markers associated with Abscission, and you do these survival curves or these rates of Abscission curves, and you also put a treatment, what if the treatment would reduce the weight as well? Do you do weight correction? Because I would assume that if say a fruit loses a lot of its weight somehow, maybe that's not happening, it will.
Fruits loses weight?
Well, it doesn't lose, but does not grow in the same level. So you apply a treatment.
So this is one of the things that we saw, that when we applied this substance, there is no decrease in the fruit weight. So we're not getting smaller fruit. And this is something that is really good, because actually we did experiment using 2,4-D, which is an auxin. We can use an auxin to mitigate or to reduce the abscission. However, with the 2,4-D, we get an enormous decrease in the rate of abscission, but the fruit were smaller. So it's really not a good thing for the grower. And with this, we are seeing that we have a possibility to reduce absorption without affecting the size of the fruit.
Oh, okay. Maybe you said that and I missed it. Okay, but that's but anyways, no.
Yeah.
Okay, I heard. Yeah, wonderful.
Okay.
And now I have another question about do you use, so when I look at these curves, you know, in the field of statistics, survival analysis, there's a Cox model. This is kind of, you're looking at time.
You will have to talk louder, Yoni. You will have to
Okay,
talk louder.
I will get. Yeah, really?
Yeah.
Okay. So do you use survival analysis tools such as the Cox model or other statistical tools, in the sense of you look at time to event models? So you make a statistical model of the time to event.
Yeah.
Has there been a need for that?
Is there been?
Have you had a need for that? For more complex statistical models?
No, we haven't used more complex statistical model, because we are just counting the fruits. We're going to the field or in the fruitlet explant. We're counting the fruitlets that we have in our system or in tag shoots. And there we're coming to the fields in various interval and counting what is remains on the fruitlet, on the shoots. And then we are simply calculating the abscission rate, and we're doing simple statistical analysis to see whether it's significantly higher using ethaphone as compared to control using student T, simple. But no more complex than that for our purpose, because just we want to see that when we're sampling, or what we are checking is before the abscission reaches the peak, because we want to see what is occurring before the abscission. This is exactly the idea.
Got it. Okay, fair enough. Maybe that's needed.
Okay.
And just a final question. Sorry, maybe it's very hard for you to hear me.
No, I can hear you perfectly. Can you speak?
Okay.
Now you're freezed. Yoni? Can you speak, please?
Yeah, I hear you. Yeah, so just a final question. Is there a situation where abscission is something that you know, here you want less abscission, but is there a situation where there's you've reduced abscission so much that it's actually negative on the production? Is there such a case, or is it not
So
a
there is
situation?
natural abscission. There is natural abscission in order to, because the tree cannot, there is an enormous quantity of flowers, there is a fruitlet, enormous quantity of fruitlets, but the tree cannot cope with all these fruitlets. So the natural drop is something natural that occurs for the fruits to develop correctly. However, yes, if there is an increment in the abscission due to wave of heat or something like that, eventually we can this is exactly the agronomical problem. There is more abscission than the natural abscission that usually occur, and there are even cultivars that are more prone to abscission, and they can eventually get a situation in which they have very less fruit when they want to harvest it. So this is something that's really, this is the problem really. So this is why and especially with the increasing temperature nowadays, we will have to find a way or a solution to mitigate the natural abscission.
Okay. I hear you. Well, thanks a lot, Vered. In the name of the audience and me, I'll tell you thank you. I'll stop the recording here, and anybody who wants to get in touch with Vered, you can go to just search for her through the Volcani Institute or the link from this recording's website, and please do, because she is also seeking further collaboration with Australia, and there's a lot to do. So thank you on that. I'll just stop the recording.
Thank you very much. Bye.
Thanks. אוקיי, אני רואה שאני לא